Hi folks, my name is Cole, and today we're going to discuss a paper that talks about naive T-cell editing in live mouse models. Now, the paper itself is by Nüssing et al. And is called Efficient CRISPR-Cas9 Gene Editing in Uncultured Naive Mouse T-Cells for In Vivo Studies. Before we start to look at what the paper describes, I'd like to share just a little bit of background information to give some context to all of this, especially because the paper itself deals with the problem of generating gene-deficient cells in organisms for molecular pathway study. To start off, when looking at specific genes, you have a gene of interest. In a mouse model, this gene of interest is completely functional. To look at what this gene does, we often knock it out to see the effects that it has on the organism. There are two ways of doing this. So you can make a constitutive knockout mouse, which has your gene of interest turned off at all times in all tissues, so there is no expression of this protein. Or you can create a mouse where this gene is turned off in specific areas and at specific times, so you can see how it functions in these places. Creating these mice is a laborious process that takes months to years to get fully stably expressing knockouts. This is done by isolating a gene of interest and putting it into cells, having those cells grow up into colonies that you screen for. These cells are then implanted into mouse embryos, which grow up to be chimeric pups. These chimeric pups are then bred back with the wild type. These then create F1 heterozygous pups for your gene of interest to knock out. These pups then need to be bred again with each other to create your gene of interest that is knocked out in the organism. Now, there are many different cell types that you can edit in this way. One population of cells that you can edit in this way are immune cells. And immune cells are grouped into two basic categories. You have your innate response cells, which create fast-acting, non-specific responses versus pathogens and infections. Or you have your adaptive response cells, which take a little bit longer to initiate, but have more specific cell responses to pathogens and infections. The cell that I'm focusing on today are T cells, and these are cells found in the adaptive response cell grouping. There are two different types of T cells. You have your CD4 T cells, which there are many types of, and can help activate or dampen immune responses. But what I'm going to focus on today are CD8 T cells. And these cells are cytotoxic and kill other targeted cells. These cell populations expand by doubling DNA and breaking it off into the sister cells. Now, this is not the only way that DNA can be broken. It can also be damaged as a result of radiation, chemicals, or different enzymes to create double-stranded breaks in the DNA. These double-stranded breaks are repaired by two different mechanisms that the cell employs. The first mechanism is non-homologous end joining, which creates an insertion or deletion mutant, which leads to frame shift of the DNA, which knocks out target genes. You also have homology direct repair, which utilizes donor DNA to create precise alterations in the DNA sequence, which can lead to gene knock-ins. One technology that utilizes this form of editing is CRISPR. This is a really interesting technique that uses guide RNA, which is complementary to the DNA, to bind to your target DNA sequence of interest, which recruits the enzyme Cas9 to create a double-stranded cut in the DNA. And the cell then utilizes its DNA repair mechanisms to create a mutation in the DNA that you can use for further analysis. Both the Cas9 enzyme and the single guide RNAs can be delivered separately. However, they can also be delivered together in something called a ribonucleoprotein, which is just RNA binding protein and RNA together. CRISPR is such an important technology that we can utilize now. This was discovered by Emmanuel Charpentier and Jennifer Doudna in 2013. And its importance was so profound that in 2020 they were awarded the Nobel Prize for Chemistry, which is fantastic. I mentioned how there are two components for delivery. And in previous studies of T cells being edited by CRISPR, or they delivered these components by either viral delivery mechanisms or by electroporation in of plasmids. Upon component delivery, these cells are then screened to identify the knockout cells. An important question to ask of all science is, why is this important? This particular paper is important because it helps improve upon a technique for further study. By improving on this technique, we can hopefully use it to better understand mechanisms of cellular pathways. So now that we have some context for this paper, we can start getting into what Nüssing et al. Found in the paper itself. And again, this paper challenges gene-specific knockout practices and particularly challenges whether T-cell activation is required for efficient CRISPR editing of cells. So what they found is that in 2 different T-cell models, they were successfully able to put their ribonuclear protein, which is a combination of single guide RNA and Cas9, into T cells and activate them with viral stimulation to successfully knock out different receptors on the cell surface. To ensure that these cells are functioning the way they should be after editing, they also checked whether the cells had similar non-target protein expression to unedited cells. And they found that this was true. There was no significant difference between different cell receptors, whether they had been edited or not, which is pretty great. Like I had mentioned, they then challenged whether T-cell stimulation was actually required for this. They found that when they put their RMP complex into cells and stimulated them with virus or didn't do that, they were still able to efficiently knock out cell receptor expression. Nüssing et al. Wanted to take this even further and looked at multiple applications of this discovery and found that they were able to knock out more than one receptor at a time and were able to class switch receptors. So to sum up succinctly what they found that their single guide RNA Cas9 ribonuclear protein, or just RMP, delivery is efficient in different T cell models. They found that editing of cells does not affect their basal function. They also found that their RMP does not need stimulation to function correctly and knock out different receptors. And finally, they found that this RMP delivery can be used for multiple receptor knockouts and point mutations. This information is important to have uncovered because it gives us tools to better understand molecular pathway analysis. And by being able to better uncover these pathways, it can lead us to new technologies that might be able to help us improve treatments to different diseases such as cancer, because tumor microenvironments depress activation of cells. So if we can turn on T cells to help fight the tumors without having to activate them beforehand, it could help improve their function, and it can be used to hopefully improve cancer treatments in the future. Now, all scientific discoveries lead to more questions due to the uncovering of new information. These can be further explored by additional experiments. When reading this paper, I had a few ideas of what some of these additional experiments could be as well. So because they only looked at CD8 T cells, I was curious if this technique could also be applied to CD4 T cells. Additionally, because they were able to edit these T cells, perhaps these T cells could be used in an immunotherapy model for helping clear tumors. And finally, because they found that they could class switch different receptors, I thought this technique could maybe be used to help prevent organ graft rejection by editing the specific receptors on these cells. Now, these are just what I thought about. There are, of course, always more questions to be asked. Ultimately, though, I hope that you learned something and had fun doing so. If you are interested about any microbiology or immunology topics and want to hear some more about them, let me know in the comment section below. As well, in the description, there's a link to the paper itself if you wanted to look at the supporting data that Nüssing et al. Has provided. And thanks for watching.